Okay, can somebody confirm you can hear me?
I can hear you.
Great. Should be seeing my screen now. Before we begin, are there any questions? All right, I'm not hearing anything. So today is August 1st, and we are supposed to be doing lab 6 on disc diffusion and antibiotics. Lab 6, disc diffusion, evaluation of disinfectants, antiseptics, and antibiotic effectiveness. There is some reading to do before you begin the lab. We do have a virtual lab as well as video clips as supplemental materials in this lab. The learning objectives for this lab are: 1, identify the type of chemical agent and active ingredient used in various disinfectants or antiseptics. 2, evaluate the effectiveness of disinfectants or antiseptics using the disc diffusion method. And by disc diffusion method, I mean the Kirby-Bauer method, or the Kirby-Bauer disc diffusion method. Any question about that? All right. 3, determine the mode of action of various antibiotics. 4, evaluate the effectiveness of various antibiotics using the disc diffusion method. 5, list the variables that influence the antibiotic disc diffusion susceptibility testing, meaning when we're using the disc diffusion method, there are some variables which can influence how effective the antibiotic is. And you need to be able to list those variables. And then 6, define the terms. What's sterilization, disinfection, a disinfectant, an antiseptic? What's a bactericidal agent, a bacteriostatic agent, an antibiotic? What's a spread plate? And what's the zone of inhibition? Any question about any of those learning objectives? All right, so let's move on to the evaluation of disinfectants and antiseptics. The terms sterilization refers to the elimination of all viable organisms, including endospores. There are no degrees of sterilization. Either something is sterile or it is not. Now, when we are talking about sterilization, we are not talking about the complete removal of viruses or, let's see if I can get that term right, prions, proteins, prions, but we are referring to the removal of all living cells, including endospores. How we typically sterilize something in microbiology is we put it in the autoclave, and that will kill even endospores. Disinfection refers to the destruction of vegetative cells, not necessarily endospores. We usually use a chemical agent that we call a disinfectant to engage in disinfection. These chemical agents are then applied to inanimate objects to remove vegetative cells, but not endospores. So not all disinfectants kill all endospores. And even some disinfectants that do kill some endospores do not kill all endospores. But there are some disinfectants, and this is kind of confusing, I know. There are some disinfectants like hydrogen peroxide, which will kill all endospores. But there are other agents, which are a disinfectant like bleach, that will not kill some species of endospores. They may kill some species of endospores, but not other species. And a chemical which is used on an inanimate object is called a disinfectant. If that same chemical is used on living tissue, we don't call it a disinfectant, we call it an antiseptic. Usually, antiseptics are less harsh chemicals that are less irritating to the skin than a disinfectant. But, for example, you could use the same chemical on an inanimate object, like using bleach on a toilet. The bleach is a disinfectant. And then your cat comes home with a torn something that's bleeding, and you want to sterilize it, and you don't have anything to sterilize it with, so you use bleach. You're using the bleach then as an antiseptic. Any question about any of that?
So I have a question. Is it not true then that when you're disinfecting, you're sterile? I mean, they're not hand in hand. Sterilization is not the same as disinfectant, correct? Like I'm understanding that correctly?
That is correct. When you disinfect the toilet with a bleach, you are not sterilizing the toilet. All of the endospores that are there will continue to be there. Well, not all of the endospores. Most of the endospores, I can't even say that. Many of the endospores that are on the bleach, on the toilet, will remain there with exposure to bleach. Some of the endospores will be killed, but not all of them. There are some exceptions, like if you were to sterilize your toilet with hydrogen peroxide, that would kill all vegetative cells and all endospores. So actually, hydrogen peroxide would be one disinfectant which would sterilize the object you're cleaning. But most do not. Most disinfectants do not destroy endospores. All right. To evaluate the effectiveness of chemical agents, the surface of a nutrient agar plate is inoculated with a particular bacteria. Paper discs saturated with different chemical agents are then applied to the surface of the agar. The chemical agent will then diffuse into the agar, establishing a concentration gradient around the disc. Next to the disc will be a higher concentration. Further out from the disc will be a lower concentration. You then incubate the plate overnight at 35 degrees C, and then observe the bacterial growth pattern around the disc. And you're looking for a zone of inhibition, an area with no bacterial growth. The chemical is assumed to be effective against that particular bacterium. That doesn't mean that the chemical or that bacteria is sensitive to that chemical or resistant. All I'm saying is that chemical gives a zone of inhibition. Conversely, if no zone of inhibition appears, the chemical is assumed to be ineffective. And if there's no zone of inhibition, you could say that the bacteria is resistant to that chemical. But sensitivity and resistance mean something different than simply whether there is a zone of inhibition or not. Is that clear to everyone? All right. So this table is looking at the different chemical agents, the general action, and then the preferred use, like chlorine bleach is used as a disinfectant. A tincture of iodine is used as an antiseptic. Any question about any of that? Where do we have hydrogen peroxide? Used as an antiseptic. And Like I said, this one is one of the antiseptics or disinfectants that we're talking about, which actually does kill endospores. Most of these others do not. There is one that does. Let me see if I can find it. I'm not seeing it. Here it is here. The aldehydes do kill endospores also. Oh, good. My computer will speed up now. I just finalized making the lecture Zoom page. The zone of inhibition size depends only partially upon the sensitivity of the organism to the disinfectant or the antiseptic. There are various other factors that affect the zone size, including the concentration of the chemical. Generally, the greater the chemical, the larger the size of the zone of inhibition. The molecular weight of the agent also affects the zone of inhibition. Generally, the larger the molecular weight of the agent, the smaller the zone of inhibition. And the density of the bacterial inoculum also affects the zone size. The reason for the molecular weight affecting the zone of inhibition is the larger the disinfectant, the less it will diffuse through the agar. And so the larger it is, the smaller the zone of inhibition. Therefore, we have to standardize some of these variables when we're evaluating the effectiveness of the agent, such as an antibiotic. Antibiotics are actually any antibacterial agent can be said to be bactericidal if they kill the bacteria. If they don't kill the bacteria and they only inhibit the bacteria's growth, we call it a bacteriostatic agent. Some antibiotics are bactericidal, other antibiotics are bacteriostatic. And don't ask me which is which, because I don't know most of them. I think most antibiotics are bactericidal, but I could be wrong on that. Penicillin G is bactericidal. I know that one. When bacteriostatic antibiotics are used, we are relying on the immune system to assist in the elimination of the infection, because a bacteriostatic antibiotic will only inhibit the growth of the microorganisms. And once the bacteriostatic antibiotic is removed, the organisms may then regrow. And so we rely on the immune system to eliminate the infection. Any question about any of that? All right. The disk diffusion method is also called the Kirby-Bauer disk diffusion method, which is a test most commonly used for testing antibiotic. And it's named after, obviously, two doctors who invented the method, Dr. Kirby and Dr. Bauer. The results of the Kirby-Bauer disk diffusion test can help a physician choose which antibiotic to treat a patient with. The bacteria can be isolated, made into a pure culture, put onto an agar plate, and then filter disc impregnate the plate to test the antibiotic against the bacteria. You then incubate the plate and then look for a zone of inhibition. Where the concentration of the antibiotic is high enough, we will get a zone of inhibition, which will inhibit the bacterial growth. It may inhibit the bacterial cells from growing, or it may kill the cell, depending if it's bacteriostatic or bactericidal. The inhibition zone size is dependent on several factors, such as the diffusion rate of the antibiotic. A smaller antibiotic will give rise to a larger zone of inhibition than a larger antibiotic. The degree of sensitivity of the microorganism. An organism which is more sensitive to that antibiotic will have a larger zone of inhibition. The growth rate of the bacterium. Bacteria cells, which are slowly growing or not growing, will be harder to kill than fast-growing bacteria cells. And so a slower growing culture of bacteria will have a smaller zone of inhibition than a fast-growing culture of bacteria. Any question about any of that? Okay, table 6.2 lists the procedure protocols required to standardize the variables that can influence the Kirby-Bauer disc diffusion test. So first, you inoculate a broth using several bacterial colonies isolated on a non-selective media. And they are no more than 24 hours old. You grow the broth until the turbidity of the broth reaches 0.5 Macfarlane units, and that will be approximately 1 × 10⁵ cells per mil. If the broth is higher than that, you dilute the broth, and if it's greater than that, you'd have to continue growing. No, if it's higher than that, you dilute the broth. If it's less than that, you have to continue growing the broth, the culture. You inoculate the broth onto Mueller-Hinton agar plates at room temperature. We always use room temperature and Mueller-Hinton agar plates between pH 7.2 and pH 7.4, because at different pH, you can get a different zone of inhibition size, as well as at different media, you can get a different zone of inhibition size. So we always use Mueller-Hinton agar plates, pH 7.2 to 7.4. The agar depth is always 4 millimeters, because the deeper the agar, the slower the antibiotic will diffuse through it. And obviously the shallower the antibiotic, the quicker the antibiotic will diffuse. So the depth of the agar is important, as well as the size of the agar plate. We always use plates which have a diameter of 150 millimeters. We always inoculate the plate using a non-toxic swab, using the spread plate technique to obtain a lawn of growth that's confluent growth on the plate. And I'll show you that in a minute. You then place the plates, place disc on the plate 5 minutes after applying the bacteria, but no longer than 15 minutes after applying the bacteria. You then incubate let the plate sit for 5 minutes, and then add the plate to the incubator, a non-CO2 incubator, for 16 to 18 hours at 35 degrees Celsius. 15 minutes after adding the antibiotic dis. I think I said 5, I meant 15. If you incubate the plates for greater than 16 to 18 hours, you'll get a different zone of inhibition size, as well as you incubate the plates at a different temperature, you'll get a different zone of inhibition size. Let me see if I can remember this. If you grow for a longer period of time, I believe the zone of inhibition will be smaller than if you only use 16 to 18 hours. And it really depends on the species, but if you grow at a higher temperature, well, it depends on the species, I guess. If you grow at a higher temperature, the bacteria will be growing faster for some species, in which case the zone of inhibition size will be larger. But if you grow it at a temperature where the bacteria grow slower, the zone of inhibition will be smaller. All right, so those are some of the variables affecting the Kirby-Bayer diffusion method, affecting the zone of the inhibition size. After incubation, the incubation sizes are then measured in their diameter using millimeters. You then compare the measurement to a table set up by the National Committee for Clinical Laboratory Studies to determine whether the zone of inhibition of the bacteria is sensitive, intermediate, or resistant to that particular antibiotic. So there's a zone of inhibition, and we would measure that using millimeters, compare that to the table set up by the NCCLS, the National Committee for Clinical and Laboratory Studies. And if the zone of inhibition is greater than the table says it's sensitive, then that will be sensitive. And if the zone of inhibition is smaller than the size that the table lists for resistant, we will call that resistant. Any question about any of that? So determining whether a species is sensitive or resistant to a given antibiotic requires setting up the Mueller-Hinton-Auger test under the standard procedure, growing the test or the bacteria on the plate for a certain number of time, looking at the plate, and then measuring the zone of inhibition in their diameter and comparing that measurement to the table set up by the National Committee for Clinical and Laboratory Studies. And in this case here, let's see what this antibiotic is. Staphylococcus aureus. For the antibiotic penicillin G, the diameter is 20 millimeters, which means that this organism, Staphylococcus aureus, is resistant to penicillin G. Any question about that? All right. So that's how we determine if a species is sensitive or resistant to the antibiotic. And you can note that there's a third category where we say that the species is intermediate. That will be between 20 millimeters and less than 29 millimeters. The species would be intermediate in their resistance or sensitivity to this antibiotic. The table will be different for each antibiotic and for each species. Any question about that?
What does the intermediate mean?
It means the size of the zone of inhibition is not great enough to be classified as sensitive. This organism is not sensitive to that antibiotic. However, the size of the zone of inhibition is greater than the size that the table says is the bacteria is resistant to that antibiotic. Is that clear? So this, if it were to be 1 millimeter longer, for example, the diameter, then it would be intermediate. It is neither resistant to this antibiotic or sensitive to the antibiotic.
So in essence, would it be ineffective? Like, would it not work as well? I guess
Well,
I'm just
it wouldn't
confused.
work as well as a antibiotic where the species would be sensitive to it. Okay,
Okay, thank you.
it wouldn't work as well. I'm not going to say it's not going to work at all because obviously there is some zone of inhibition. So it is killing cells. This is penicillin G, so I know it's bactericidal. So it is killing cells. But then you could even argue that if it's 20 millimeters like that, the penicillin G is killing some cells. It's just not killing them beyond 20 millimeters. And so we would prefer not to use this antibiotic because we classify this species as being resistant to that antibiotic. And so if you were to treat this patient with penicillin G, any bacteria that would be 20 millimeters or away from the blood vessels, those bacteria would not die. Okay? All right, know the terms of this lab. And then perform the laboratory exercise. You're not going to be physically performing any of these activities, but you will be held responsible for how to interpret the effectiveness of antibiotics using the Kirby-Bayer disk diffusion method. And let's go to the worksheet now. Not quite. For procedure 1, we have preparing a spread plate, and you're going to watch this video clip on preparing a spread plate. You can review the steps using aseptic technique when you're preparing a spread plate to obtain a bacterial lawn of growth. And let me go ahead and just detail that a little bit. Pretend this is a plate with agar in it. You get your cotton swab. It's obviously sterile. Dip it in your test tube containing your stock culture. Put it in the tube. Get the swab wet, and you don't want to get it dripping wet. So what you do is on the inside of the tube, touch it against the inside of the tube. I'm going to show on the outside because I don't want to put the pencil, which is not clean, in my drinking water. But you get the worst of it off by touching the inside of the tube. And then you swab everywhere with the cotton swab. I don't know if you can see this. I'm trying to show that I'm swabbing everywhere.
Can see it better now.
Okay. And then you rotate the swab a quarter of a turn, rotate the plate a quarter of a turn. And once again, you swab everywhere, trying to get this so you can see it. I'm not going to be able to swab everywhere because I've got it at an odd angle for showing this. Okay. And then you rotate it again a quarter of a turn, rotate the plate a quarter of a turn, and then swab everywhere one more time. You need to swab everywhere because I once had a student who simply went do, rotate, rotate, do, rotate, rotate, do. And she had huge areas of the plate, which she did not swab. And so there was no bacteria in huge areas of the plate. And then she showed it to me and said, how can I measure the zone of inhibition? And I said, well, you didn't swab the plate everywhere, so you cannot measure the zone of inhibition. You will either have to repeat the experiment or you can use your lab partner's plate, because on her plate, it was totally impossible. You need to swab everywhere. And after you swab 3 times, you then take the cotton swab and then wrap it around the edge of the plate. Trying to show you that. Because when you swab from side to side, you may miss the very edge. And so we swab the very edge of the plate to get the bacteria to grow everywhere. And then the bacteria should look. Oh, we don't have a picture. Well, kind of like this. Let me blow this up so you can see it better. Where the bacteria will be growing everywhere on the plate, except for in the zones of inhibition. And that's because we swabbed everywhere on the plate. And that we call a lawn of bacteria, where the bacteria is growing everywhere, sort of like grass is everywhere in your lawn. Any question about that? Oops, now I'll shrink this down. Someplace around here. For procedure 2, you're going to evaluate the antibiotic effectiveness. You can view the following video clips and complete the virtual lab. So that will be a video clip preparing a culture for Kirby-Bauer diffusion method, and then the interpretation, a video clip on the interpretation of the results for the Kirby-Bauer diffusion method, and then a link to the virtual lab. Let's see if this works. I'm not sure that's the new link. It is. Okay, great. This link may take some time to open and download on your computer. You need to hit the start button. And how you work this is you can hit the menu there, and you take this ruler and you take it to the edge of the zone of inhibition. I don't quite have that on the edge. About right there. And then measure the zone of inhibition. So if you look there, that's about 18 millimeters, maybe 19 millimeters. And that's CP, which stands for, I'm not reading that right. SCF, sorry, CF, which stands for cephalothin. And what did I say, 18 or 19? So that is sensitive. And if ever you get the click there and it's green, that means it's correct. If you click and it's an X, a red X, that means it's the wrong selection. The same with that one. All right. And then for erythromycin, I don't even have to put it there because there's no zone of inhibition on E for erythromycin. So we'd say the zone of inhibition is less than 13 millimeters. So once again, it's sensitive. I'm going to get the next one wrong. Oxacillin, I'm going to say it's 11 to 12, and it's an X. 13, also an X, so it's less than 10. the zone of inhibition. All right, so that's how you do this. To get the different plates, you just click the menu button. So E. coli is the first plate. Click the menu, restart, and it'll go to Pseudomonas aeruginosa. And there's the zone of inhibition there. Just bring the ruler up there. I'm going to move down a little bit to try and get that. Someplace like a round right there. For ciprofloxacin, on many of them, it's nothing on this one. And then there's a third plate. Just hit the menu button again and restart. And it's Staphylococcus aureus. It looks like only vancomycin is the only one which has a zone of inhibition here. And its sensitivity is about 17. So it is sensitive to vancomycin. And I'm going to guess this is MRSA, multi-drug resistant Staphylococcus aureus, because it's resistant to all of the antibiotics with the exception of vancomycin. Any question about any of that? Anyways, there's 3 plates. To go back to the very start one of the E. coli, you can just do it 3 times. Any question about that? All right. Yeah, I can close this. And then 2, use the standards for Kirby-Bauer distaffusion susceptibility test on E. coli. Using the spread plate technique, antibiotic discs are placed on the surface of the agar and incubated overnight. Below are the results along with the NCCLS zone standards used for interpretation. And then let's go to the worksheet here. Remember, only submit the worksheet. If you submit the lab module, you may get a penalty. Because if everybody were to submit the worksheet, my Canvas website would soon become filled, and then nobody could submit their assignment. So, answer question 2. We have the Kirby-Bauer distaffusion test, and there's one there for the antibiotic ampicillin, which is the bacteria. I think it's E. coli. Yep, E. coli. And there's the zone of inhibition measured there. And then the NCCLS recommended standards for which are resistant and which are sensitive zones of inhibition for the antibiotic, for each given antibiotic. And then we have ampicillin here, cephalothelin there, gentamicin here, and tetracycline there. So what you do is you measure the size of zone of inhibition. I've got the first one filled in for you, ampicillin. The measurement of the zone of inhibition is from this 0 to that 0, or very close to 15 millimeters. You do not measure from here to there, which would be close to 10 millimeters, because if you notice, the 0 mark here doesn't begin here. So we don't measure from here to there. We measure from 0 to 0. Any question about that? That's how you use the calipers. You measure from 0 to 0. And the first one is very close to 15 millimeters, which I've got put in there. And then you use the interpretation of the table for ampicillin. And 15 millimeters will be this result right there. Any question about that? And then you measure for each of the other different antibiotics. 0 to 0 there is about 14 millimeters for cephalofluffen. You put that there and then make your interpretation. Any question about that? All right, so in this table, you must fill in the zones of inhibition size in millimeter and then give the interpretation. Whether it's sensitive, you can just write an S, or intermediate, an I, or an R for resistant to these antibiotics. Then answer question 3, list 5 variables that must be standardized when performing the antibiotic susceptibility test using the Kirby-Bayer disk diffusion method. And you can get that from a table in the lab module. And then 4, don't ask me why I have that twice. The size of the zone of inhibition often varies for 2 different species of bacteria when challenged with the same antibiotic. What is a possible explanation for these differences? Answer that question. And then move on to 5. What would happen if we didn't use aseptic technique to put the antibiotic disc on the plate? Mention what will happen to our test results. Like if you don't use aseptic technique, you know the plate's going to become contaminated. And how may that affect the test results of our test? 6, what would happen if we increase or decrease the concentration of antibiotic in the disc? Be specific in your answer. And your answer has to explain what happens when we increase the concentration, as well as what happens when we decrease the concentration of the antibiotic. 7, antibiotics can be bactericidal, meaning they kill the microbe, or they can be bacteriostatic. They inhibit microbial growth, but do not kill the microbe. When observing a zone of inhibition around a disc, how would you determine whether the antibiotic causing that zone of inhibition was bactericidal or bacteriostatic? So right here, we have a zone of inhibition. And how can you determine whether that zone of inhibition is from the antibiotic being bactericidal or bacteriostatic? I'll warn you that this question is a thinking question, and it's quite challenging for some students. Many students get it wrong the first time. However, the second time, most students get it right. So this is a thinking question, but you should be able to answer it. All right, 8. When performing antibiotic sensitivity testing, it's important that the test conditions are kept very standardized, meaning we use the same kind of media, Mueller-Hinton plates. We use the same agar depth, 4 millimeters, and the same age of the culture. Why? And 9, are there any other substances besides antibiotics that we could evaluate using this method, meaning the Kirby-Bayer disk diffusion method? And you should state one other substance. You can either name it directly or you could name it indirectly, but you should name one other substance. Okay? For example, if you thought hair tonic would cause zone of inhibitions around the hair tonic, I'm just saying something off the top of my head. Don't ask me why I thought of hair tonic there. If it were, you could say hair tonic Or you could say a specific hair tonic, like, I don't know, Grecian Formula 12. You just need to name one thing. Okay. And I gave you one that, to my knowledge, does not form a zone of inhibition. So it's not one you can use. You have to come up with something. All right, 10. Does the presence of a zone of inhibition necessarily mean that the bacteria being tested is sensitive to that antibiotic? Explain your answer. And then 11. From the size of the zone of inhibition of chloramphenicol on E. coli in the virtual lab, is the bacteria sensitive, intermediate, or resistant to that antibiotic? And then for chloramphenicol on the bacteria Pseudomonas aeruginosa, is the bacteria sensitive, intermediate, or resistant to that antibiotic? All right, that's it. Any questions? If not, I will be here in the lab until 9 o'clock or the last student logs out to help you with the lab and answer any questions. So if you want help or you have a question, remember to ask the question early, because once the last student logs out, I too will log out. Any questions?
I have a question. On the worksheet, it starts at number 2, but in the lab module, there's procedure 1 with a question. Is that correct?
I don't know. Let me look into it. So the lab module will be different than the lab worksheet. That is true. Okay. Is that what your question is? Or are you saying that my lab worksheet differs from your lab worksheet? What's your question?
My question was, I just wanted clarification because on the worksheet, it started with question number 2. So it seemed like there was missing question number 1.
From the lab module?
No, from on the worksheet.
I know. Yeah. Follow the worksheet. We'll just go with that, okay? I'm not sure where there's question number 1. Is the question number 1 not even present?
Correct.
Yeah, then just, there is no question number 1. It started with question number 2. And that's just the way that the person who sent it to me did it, and I just kept it, okay? So originally, there probably was a question number 1, and my guess is that the person who designed this worksheet removed it. He didn't like it or he didn't want it, and he didn't restart the numbering. Okay. So
Okay,
that's
thank
why
you.
it starts with question number.
I just wanted to double check. Thank you.
Okay. Initially, there were 2 question number 2s right there. And then I think number 5 was question number 2. And I just renumbered them. All right. Let's stop the video.